So there is going to be a lot of terminology that we're going to be throwing around. And one of them is the idea of the sense strand and the antisense strand of DNA. The sense strand is what your eventual coding portion of DNA is going to look like. And the antisense is going to be the mirror copy of the sense strand. And what is kind of confusing about this is that the antisense strand is actually going to be the template that the transcriptional machinery is going to use to create your RNA. The sense strand is what's going to be what the eventual coding RNA is going to look like. And so sense strand is not actually used by the transcriptional machinery, the machinery that actually copies the DNA into RNA to make the RNA, but that's what the actual sort of RNA is going to eventually look like. And so we've talked a lot about DNA versus RNA. So the question is then, what is the real difference between DNA and RNA? There are a couple of differences. The first is that RNA is generally single-stranded. It doesn't pair with any other copy of RNA, whereas DNA is double-stranded. It tends to form base pairs with a second strand in the nucleus, or with each other, sorry, not necessarily in the nucleus. The other thing is that there is different actual functional parts of RNA. So you've heard of AGCT, hopefully. Those are the nucleotides that code for your DNA. Your RNA has the same A, G, and C, but rather than having a T, it has what's called uracil, or U. So those are sort of two of the differences that we see between DNA and RNA. Single-stranded RNA, double-stranded DNA, thymine in DNA, uracil in RNA.